Welcome everyone to our talk today. So my name is Nghia. I'm the coordinator for today, and there are the two speakers we have, Moon and Deepanshi. And as you can see from the slides, this talk will guide you through the chocolate factory of life, from DNA recipes to protein packages. But first, before all of this, I will do some introductions on the speakers. So the first speaker we have is Sue Moon. She's a first-year PhD student from the cell biology department, and her research is on how fats move within cell. A fun fact of hers is that she has traveled 25 countries and lived in 7 cities so far. And the second speaker is Deepanshi. She's a fourth-year PhD student in the chemistry department, and her research is on studying protein motions using nuclear magnetic resonance, which is a big magnet that she will talk about later on. And a fun fact of hers is that she can blow glass. So to start off with our discussion for today, we can talk about the cell. So the cell is something that everyone knows about, right? It's the fundamental unit of life, is something that is very essential to all life. And here is a typical rendition of the cell with all of its compartments inside of it. And a question I want to ask people maybe is, what does the cell do? And when you think about this, the cell actually have a very big function in making stuff, making things for the body. And so to understand the function of the cell and all of its compartments, we can have a really useful analogy of the cell to a chocolate factory. And in this sense, then, the cell is a factory that produces chocolates in all different flavors, different sizes, different properties that are sent to customers throughout the body. And in this sense, too, the compartments inside of the cell here are actually very analogous to different machines inside the factory line. And I think what's exciting here is that this analogy is not very far off from reality, because in reality, even though these molecular machines are very small, they are also very precise, they are very dynamic, and they serve very specific functions. So for example, the cell has its own motor that you can see is moving very accurately inside of the cell. The cell also has a scissor that cuts DNA. And then the cell also has this little guy here working around transporting cargo. And so this is what our two speakers today will be talking about. So first, Su Moon will give a tour of this chocolate factory, and she will talk about how the cells produce, package, and ship a type of product called proteins. And then our second speaker, Dipanshi, will zoom in and study how cellular machines work by listening to its molecular whispers using the NMR, which is the very big magnet that she will be exploring.
Hello, everyone. My name is Sue, and I will take off from Nia's part and share my part of the presentation. If you guys have any problems in hearing or seeing the screen, then please tell us.
Please enter.
So, hello everyone again. Welcome to the Chocolate Factory tour. My name is Sue, and today I will guide you through how proteins are made and transported. So, to understand the chocolate factory of our body, let me introduce an example of insulin production and diabetes that is related to this topic to better understand. Here is an adult male body with diabetes. Our body is considered as the largest scale in the levels of biological organization. And the endocrine system is one of the major organ systems of our body. It controls and coordinates important body functions, but more slowly and with longer lasting effects than the nervous system. And each organ system is composed of multiple organs such as stomach, heart, or liver. And the pancreas is an organ that is part of both the endocrine and digestive systems. Each organ is organized into multiple layers of tissues. The pancreas has tissue called the pancreatic islets. And within these pancreatic islets, there is a specialized type of cell called the beta cell. Since we are talking about humans, beta cells share the general characteristics of an animal cell. And plants have a different type of cell. And in an animal cell, there are many organelles, membrane-enclosed structures that carry out its unique functions. And today you're going to hear a lot about organelles, especially the nucleus, the endoplasmic reticulum, or in short, ER, and Golgi apparatus, or in short, Golgi, and lysosomes. These organelles are involved in creating, discarding, modifying, or transporting macromolecules. And the organelles within these beta cells work together to produce insulin molecules. Insulin is a small protein that regulates blood glucose levels. And if we cannot make insulin properly, our body will not be able to regulate blood sugar levels. So, low blood sugar levels can result in dizziness, sweating, trembling, or irregular heartbeats. And high blood sugar levels can lead to diabetes or kidney failures. So, insulin that regulates blood sugar level is a type of macromolecule called proteins. And proteins are very important because they help our body do many essential functions. For example, first, proteins are important building materials for our body, helping form cells, tissues, and organs. They help repair damage, like when your body heals after a small injury. And second, proteins help make hormones, which control many processes in our body and keep it balanced. For example, they regulate things like energy levels throughout your entire day. Third, proteins help control how our genetic information is used inside the cell. They help decide which instructions in our DNA are turned on and off. And as you can see, our body cannot function without proteins. Therefore, it is important to properly make and transport those proteins to correct destinations. So, how does our body do that? Cells make, package, and deliver proteins through a pathway called the protein secretory pathway. And this process is similar to a chocolate factory, how chocolate is being made, packaged, and delivered to each customer. Think of proteins as chocolate. So this is the overview of the secretory pathway of proteins. First, organelles called the nucleus and the endoplasmic reticulum is responsible for making proteins, similar to how the chocolate factories make chocolate. Second, proteins move to the Golgi apparatus, where proteins are added with chemical tags. It is similar to how chocolate is packaged and added with address labels. Finally, the vesicles transport the packaged proteins out to correct destinations, either in or out of the cell, just like the delivery trucks. So now we will go over each step of this protein secretory pathway and discuss about what machines play a crucial role here. And of course, we will continue our tour in the chocolate factory too. So before we begin making chocolate, we have to receive orders. These orders come from small molecule called ligands. These ligands bind to special structures on the cell called the receptors. And you can think of this process as a key fitting into a lock. Cells receive signal from outside the body, just like how factories receive chocolate orders from individual customers. Cells can also receive signals from neighboring cells, just like how a neighboring company can ask for cooperation to make chocolate. When those ligand molecules bind to the receptor, the cells relay the signal to initiate response, which is protein production. So just like when the factory receives order from a customer saying, I want to order almond chocolate, then they start to make it. So without receiving the correct signal, the cell will not know what to make. Now that we have received the order, let's start making it. First, I would like to explain in detail what happens within the organelle called nucleus. DNA, a macromolecule that stores genetic information, is maintained and replicated inside the nucleus. DNA contains all of the information on when, what, where, who, and how the protein should be made. You can think of DNA as a recipe book to make the best chocolate product. To preserve this recipe book, the chocolate factory has to make multiple copies of the recipe book. And this is similar to why our body replicates DNA. And this picture is a closer look of what happens during DNA replication. An enzyme called DNA polymerases act as a glue to add new DNA building blocks based on the original sequence. And the DNA polymerases also ensure that the newly made DNA strand is the exact same copy of the original sequence. So you can think of DNA polymerases as a copy machine that photocopies the recipe book. And inside the nucleus, DNA is converted to another type of macromolecule called RNA by the RNA polymerases. This process is called transcription. And in the chocolate factory, to make the actual chocolate, you would have to read from the recipe book to make the ingredients. RNA molecules can be considered as the intermediate products in the process of making chocolate, such as the cocoa liquor. And RNA polymerases that do this job can be considered as cocoa grinding machines that finally grind those cocoa beans and turn solid nibs into liquid paste. And at the endoplasmic reticulum, machines called ribosomes are attached at the surface to convert RNA into proteins. This process is called translation. If we consider RNA as the intermediate product of chocolate, such as cocoa liquor, then we can consider the protein sequence generated here as the raw chocolate. And ribosomes can be considered as these tempering machines that solidify cocoa liquor into solid chocolate. But be careful. Our body must keep the chocolate production line running smoothly. If DNA is not properly replicated or converted to RNA and proteins, again, the protein production will stop and we will experience various fatal illnesses. And within the chocolate factory, we have to move the chocolate from the department of production to the department of sales. There are two opposite directions between departments moved by vesicles. Vesicles are small bubbles inside the cell that carries materials from one place to another. If the protein is made correctly, they use COP2 vesicles to move the protein from the endoplasmic reticulum to the Golgi apparatus. Similar to moving chocolate from the production line to the department of sales. However, if the protein has problems, then they're sent back from the Golgi to the endoplasmic reticulum through COP1 vesicles. Just like when the factory is retrieving defective products back to the production line before they're being packaged and sent out. After making sure that the protein is produced properly, the protein products are sent to the Golgi apparatus, the packaging center. At the Golgi, proteins are added with various chemical tags. For example, phosphate molecules are added by enzymes called phosphotransferases. And enzymes are like tiny machines in your body. Their job is to speed up important processes, like breaking down the food you eat so your body can use it for energy. Without these enzymes, these processes could be slow to keep you alive. And those enzymes adding chemical tags to proteins are just like how chocolate is decorated with various toppings like nuts and sprinkles, then packaged into bars and pretty boxes, and added with address labels to be ready to be shipped to supermarkets and individual households. And Phosphotransferase enzymes can be considered as those packaging machines that wraps the chocolate with printed labels. Instead of being sent to the final destination, some of the proteins at the Golgi are sorted to another organelle called lysosomes. Lysosomes are in charge of degrading and recycling the waste products using some enzymes that reside within these organelles. Those enzymes are only functional when they are located within the lysosomes. So it is important that the protein should be recycled and delivered directly to this lysosome. Just like how a factory should have a cleaning facility to discard expired chocolate and recycle ingredients to make new chocolate. Finally, the proteins are delivered to destinations inside or outside of the cell by various types of vesicles. As I mentioned in my previous slide, vesicles are small bubbles within the cell that carry around materials. And vesicles are like delivery trucks that deliver chocolate products according to their address labels added in the previous step. But our body must make sure that the chocolate is delivered to the right place at the right time. Have you ever experienced these situations where you received a wrong package, or received food which you have allergies, or your package delivery got delayed way more than expected? All of these situations made me annoying and angry, and cells experience the same thing. If a protein isn't delivered to the right place at right time, then the consequences can range from mild inefficiency to serious diseases depending on the protein's role. So, for example, when the proteins are being sent to wrong addresses, the proteins may end up being useless. When the digestive enzymes are not delivered to these lysosomes and instead sent outside into the cell, lysosomes cannot break down waste products and the enzymes end up being useless. Imagine when you delivered a shovel to clean up the snow, but instead a hammer has been delivered. How are you going to go to work despite this huge pile of snow? Same thing for cells. And if the proteins are delivered late, its purpose can be missed. When proteins that controls the division of cells arrive too late, then these processes can go wrong, and wrong cell division may result in cancerous cells because they divide uncontrollably. Imagine when the chocolate factory needs to order a new milk to make their chocolate, but the delivery got delayed for a few weeks. If the factory continues to make chocolate with that expired milk, it will cause a huge trouble. And finally, dangerous contents ending up being in the wrong place can lead to cellular damage. When digestive enzymes that are meant for lysosomes end up being somewhere in the cytoplasm, then they can break down important cellular components, such as the machines that we have talked about in the previous slides, eventually damaging this entire cell. So imagine delivering an almond chocolate to someone who has almond allergies. You don't want that to happen. So it is important to make sure the delivery trucks or vesicles deliver properly. So until now, I've talked about what the protein secretory pathway is and why it's important for the system to function properly. First, proteins are made in the cell, especially in the structure called nucleus and the endoplasmic reticulum. Then they're being sent to Golgi, where they're being packaged and sorted. Finally, vesicles deliver them to correct destinations. To help you understand this better, I use the analogy of the chocolate factory. You can think of this entire cell as the factory and proteins as chocolate. And each organelle is responsible for a specific step, just like how a department in a factory handles a different part of the production process. And understanding this protein secretory pathway will allow us to identify the causes of various diseases and give clues on how we can treat those diseases. So it is important to know about it. And along this pathway, you've learned about different enzymes that perform critical steps in this secretory pathway. And I gave you an analogy that enzymes are like these machines, such as the copy machine, the tempering machine, or the package machine. So, our next speaker, Deepanshi, will explain how scientists discovered these machines and what methods do we use to study them even today.
So I hope you enjoyed the chocolate factory tour. Now imagine you are walking through a chocolate factory. From far away, everything looks smooth conveyor belts, mixers, wrappers, workers. But if the chocolate starts coming out wrong, like too thick, burnt, or lumpy, the real story is inside these machines. As you learned in biology, cells are like factories. They take in raw materials and produce products, proteins, membranes, signals, energy, and inside these factories are countless tiny machines called enzymes. They speed up the chemical reactions so life can happen fast enough. But here is a twist: enzymes aren't one big machine, they are more like the machine parts, like the gears, hinges, belts, working together in a very specific rhythm. If one hinge moves slightly differently, the whole machine can malfunction. And some enzymes are so efficient that they can process thousands of molecules per second. So it's truly industrial scale chemistry. So naturally, we would want to look inside these machines and understand how each moving part works. But then the reality hits, which is the size. Here we are looking at or comparing the size of a human, then the size of animal cell, then organelles inside those cells, which you just learned about, which can act as your transporters, delivery trucks, etc. And then your proteins, and then inside the proteins, you have the atoms. So we are used to cameras and microscopes, but enzymes live at a scale where the parts are atoms. And atoms are not just tiny, they're unbelievably tiny. If a human were the size of the earth, an atom would be like a marble or smaller, depending on the comparison. So even our best microscopes struggle to show us atomic level details, especially when the enzyme is moving and working in liquid the way it does in real life. So if we want to understand how enzymes work, we need atomic information, not just the big outline. So if we can't reliably see the machine parts at that scale, what else can we do? So in a factory, experienced technicians don't only look, they listen. A healthy machine has a certain hum. The belt squeaks when it's slipping, a motor clicks. When it's misaligned, you can learn a lot without opening the casing. A healthy machine, now I want you to map that idea to the cells, the secretory pathway, Golgi, vesicles. That is like the packaging and shipping department of the chocolate factory, as you just learned. Products get processed, checked, packaged, and delivered. Enzymes are like the machines along that line. And just like the factory machines, enzymes sound different depending on what they are doing. Except it's not sound waves in the air, it's a signal from the atoms changing depending on their environment. So, our plan is when we can't see the moving parts directly, we listen for how the atoms report what's happening. So, how in the world do we listen to something so small? We use a remarkable technique called NMR, which stands for nuclear magnetic resonance. Now, that name sounds very complex, but the idea behind it is simple. It's all about magnets. You see this tiny blue marble or earth? It's a giant but a very weak magnet. You've probably also seen or heard of an MRI machine in the hospitals. That's a much bigger, stronger magnet used to take pictures of our insides. And the M in MRI stands actually for magnetic. Now, an NMR machine, which we use in the research lab, is like an MRI's bigger, much more powerful sibling. It has a strong magnet. In fact, a high field NMR magnet can be over 400,000 times stronger than the Earth's magnetic field. This incredible power is what allows us to tune into faintest whispers from the atoms that make up our enzyme machines. So, how do we tune in? Like the music of the atoms? How does NMR work? We have this magnet to listen, but how does that work? How do you get an atom to whisper on command? It's a bit like playing a musical instrument, and it happens in few steps. First, the core of many atoms, we call that as nucleus, act as a tiny spinning top. Normally, all these little tops in our enzymes are spinning in completely random directions, like you can see these tops in random directions in this picture. Then we place them inside our super strong magnet. The magnet makes most of these tiny spinning tops snap into alignment, all pointing in more or less the same direction. Next, we do something fun. We hit them with a quick pulse of energy, a radio wave, just like the kind your car radio picks up. The radio pulse acts like a little flick to the side, knocking the spinning tops out of alignment, like you can see this one inverted here. And now, for the magic, as soon as we turn the radio pulse off, the atoms want to relax and get back in line with the magnet. As they do, they release that little bit of energy, singing their own tiny radio signal, a whisper, a note that our machines can hear. And here is the most amazing part. Think of a guitar. The thick strings play a low note, a thin string plays a high note. It's the same for our atoms. The exact note an atom can sing depends on what it is and who its neighbors are. So, by listening to all the different notes, we get a unique musical signature for a healthy, functioning enzyme. We know it's a perfect song. Now, what if the machine changes behavior? What happens if the music changes? When a machine in the factory breaks, you get spilled chocolate, a mess on the floor, and production grinds to a halt until the repair crew comes in. The factory stops working as it should. The exact same thing happens in our body. Sometimes our enzymes, which are machines, change their behavior. They might work too fast or too slow or just stop working altogether. This can be a sign of a disease. For example, you learned from Sue's talk in many cancer cells, certain enzymes go into overdrive, leading to uncontrolled growth. The spilled chocolate of the cellular world. One crucial enzyme that has to work perfectly is called DNA polymerase beta, and its job is to repair our genetic recipe book. And here is the cartoon of the polymerase beta. That's how we represent its structure. And next is, what if there is damage and how do we repair that damage? Every cell in your body has a recipe book. This book is your DNA, and it contains all the instructions for making everything in the cells. Just like in a factory, you need to make perfect copies of these recipes to keep things running. But sometimes the recipe books can get damaged. It gets a typo. This isn't a rare event. It's estimated that DNA in every single one of your cells can suffer up to 1 million damages per day. Thankfully, we have elite crew of repair enzymes that are constantly on patrol, proofreading and fixing these mistakes. Think of them as a factory's master mechanic and quality control team. One of the most important mechanics on this team is the enzyme I just mentioned, DNA polymerase beta. Now, we would want to catch this in motion. This brings us to the importance of the motions in these repair mechanics. It's beautifully shaped, a bit like a right hand, and it moves in a very specific dance to fix our DNA. It picks up the DNA using its thumb domain, and then the fingers would bring in the building block that needs to be inserted in order to fix that DNA. And then the palm domain has the sites where the actual chemistry happens. Then it closes, making a fist. Then the chemistry happens. It repairs the DNA. It opens and it releases the repaired DNA and gets ready for another cycle. Now I have been talking about like the movement and the function. Why did we want to like learn about this pole bee in motion? The idea of tracking movement to understand function is something we do all the time. Look at these famous historic images of a running horse. Each snapshot is a static. It doesn't tell you much on its own. But when you see them in a sequence, you understand the fluid motion of running. You cannot understand how a horse runs by looking at just one picture. You need to see the movement. It's the exact same for our enzymes. A single static picture of DNA polymerase does not tell us how it fixes our genes, but by using NMR to listen to its whispers, we can build a movie of its movements. We see the horse run, we see the enzyme dance, we see how its movement is directly related to its function. By understanding the dance, one can understand the most fundamental processes of life. So we have gone from a giant bustling chocolate factory all the way down to the silent, invisible dance of a single protein. And we have seen that the underlying story is the same. Life is in motion. By learning to listen to the molecular whispers of our enzymes, we are beginning to understand the beautiful, complex symphony that plays out inside every one of our cells. This isn't just a scientific curiosity, it allows us to understand what happens when the music goes out of the tune in disease like cancer. And by understanding that, we can start to think about how we can help the repair crews work better, how we can quiet the machines that are running too loud, and how we can one day learn to conduct the sweet symphony of science ourselves. The future of medicine may very well depend not just on what we can see, but on what we can hear. Thank you.